Hi, this is Anna. We're going to be looking at part 2 of Unit 13, where we look at the histologies of the male reproductive system. Alright, so right here we're looking at the seminiferous tubule. So here you've got the lumen, okay? And then right here is going to be adventitia that is separating the various seminiferous tubules. Now in the center you will have what are called interstitial cells. So inter being between, stitium is referring to the interstitial tissue, okay? And these are going to be secreting some hormones that we're going to talk a little bit more about in a bit. All right, then you're going to have extensions of this wall coming up into these structures that we call the sustentacular cells. Here's another one right up in here, okay? The sustentacular cells are important because they create the blood-testis barrier, okay? So they're basically separating the stem cells from the sperm, which have different DNA from the stem cells because they've done crossover and meiosis and all of that good stuff, okay? So they're trying to prevent antigens from escaping back into the blood system, okay? And so they create these little walls, which are easier to interpret in a picture than what we've got right here. Here's another nice sustentacular cell right here, okay? Now, in between the sustentacular cells, you are creating what you can kind of think of as kind of like compartments, okay? And up against the wall, you have these large cells that have a lot of cytoplasm and a really big nucleus. Those are the spermatogonium, all right? Spermatogonia is the plural form, all right? So that is going to be the cell that splits, and it'll form a cell and a B cell, and one stays a stem cell and the other one will go on to the next stage of meiosis 1, okay? And when it does that, we change its name again, and now we're going to have primary and secondary spermatocytes. So here are some of those spermatocytes. You'll notice that they're smaller and they're more in the middle. Now, if we were doing a more advanced class, I'd be asking you to differentiate primary and secondary on a histology slide. I'm not going to do that to you here. I just want you to remember that all of these are spermatocytes, okay? Now, once these structures begin to elongate and the nucleus is no longer round, you see a hint of a tail forming. So here, 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 all of these are spermatids, okay? Earlier ones can be a little bit more rounded, okay? So like right here, you can see a little bit more rounded ones here. Here's a little bit more rounded. The later ones are definitely much more linear. Then in the lumen, you have the spermatozoa or the sperm, okay, that are going to continue on the pathway. All right, so here we are going to blow this up a little bit bigger, and you can see a really nice sustentacular cell right here. Now, it's hard to visualize this. All right, so there's another one right up in there, but everything is actually squished in between two sustentacular cells. So the sustentacular cells are creating a compartment, okay? Up against the wall is where you're going to have spermatogonium. So I've got one labeled there, there's one there, there's one there, there's one there, okay? And then when the one that's gonna continue to become a gamete, that will become a spermatocyte here. All right, here I've labeled secondary and primary. Again, I'm not going to ask you to differentiate between those. I think they do an APR, so you're just kind of stuck with practicing it. So if you just remember spermatocyte, I'm happy. All right, so there's some more. And then here you can see an early spermatid and a late spermatid stage, okay? All right, I like this slide because it's an electron scanning microscope slide, and they've colorized it so that you can see the different types of cells, so that you can see really nicely the sustentacular cells, which are creating these compartments, and these cells are in the compartments, okay? And then you've got down here the spermatogonium, and then you've got the spermatocytes, and then they're starting to paint the red ones, the spermatids, okay? All right, now in this particular picture, we're going to focus a little bit more on this interstitial tissue. What you will notice that it's staining more pink and less purple. So this is going to be the seminiferous tubule, and this is going to be the interstitium, okay? In the interstitium, we have really nice interstitial cells which are producing the testosterone. The testosterone, in response to a protein secreted by the cystentacular cells, is going to migrate out to the spermatogonia, where when it's concentrated enough, it will stimulate it to enter meiosis 1. Okay? All right, so we're not going to spend a lot of time on the epididymis, but what I want you to notice is you can see the lumen, you can see the pseudostratified ciliated columnar epithelium, okay? And then here are the cells, all right, and you can see the nuclei, all right. This right here is like the cilia, a stereocilia. And then here you've got a nice thick wedge of epididymis, all right, and the epididymis will undergo peristaltic contractions, which will help move the sperm along towards the tail, towards the vas deferens. All right, so now we're looking at the vas deferens, and this one, the big thing I want you to take home is how thick that smooth muscle is, okay? That's the primary dominant feature that you're going to notice when you look at this under a histology slide. So the epididymis, it's got smooth muscle too, but it's more like that thick, okay? Whereas this has got super thick. Seminiferous tubules doesn't have it, really. I mean, there are little strands here and there, but not really. So the vas deferens needs to do much stronger peristaltic contractions, basically sending the sperm along at Mach 1, all right? So it's got all of that smooth muscle in order to assist with that. All right, so your obligatory hormone slide, all right? You need to know all of these. So the name of the hormone, where they came from. We've already studied this a little bit with the endocrine system, so you should be okay. So what I want you to know with the male, with FSH, is it's going to the cystentacular cells. The cystentacular cells secrete a protein, which then concentrates the testosterone about spermatogonia. This causes indirect spermatogenesis. It doesn't directly start meiosis 1, but it makes it possible for it to start occurring, okay? And then the luteinizing hormone is coming from the pituitary, going to the interstitial cells and telling them to synthesize testosterone. And then the testosterone, in response to this, telling the testosterone to move, it will go and surround the spermatogonia and directly stimulate spermatogenesis. Okay? All right, so that's the end of this particular slideshow. So you can stop here and move on to the next thing.